You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech Health Podcast, and I have Clay Wright. He's an assistant professor at Virginia Tech, Department of Biological Systems Engineering, and we've been talking about how signaling networks facilitate plasticity and robustness in plants. So we'll go into what that means and shortly. But Clay, thanks for coming. How you doing?
Yeah, thanks for the invite to be here, Richard. I really appreciate the opportunity to chat with you today.
Yeah. So tell me, I guess in plain English, what is your work and your research involved? What's a better way to restate it? Simpler.
Yeah. So my work is really trying to figure out how plants take in information from their environment and make decisions about how to grow kind of the future iteration of leaves or stems or fruits, and how to deal with threats like pests and pathogens in their environment, and try and do that in a way that is expedited so that we can actually adapt plants faster than, say, the microbial pests and pathogens that they're having to deal with can evolve, or train plants to grow in new environments that they haven't experienced before, as they might be faced with in places where climate change is going to be strongly in effect in the near future. So really sorry, go ahead.
Yeah, well, what's an example, maybe extreme examples? How do plants appear to sense and defend themselves or react to their environments? Maybe give like a very extreme example to show people some of the possibilities.
Yeah, so just recently actually published from a group in Germany, potatoes don't grow well in hot environments. That's why they grow very well in Idaho here in the U.S. and the northern climates of Europe. But as these areas where they're normally growing just fine get warmer and warmer, the potatoes become less and less starchy. The actual tuber, the potato that we eat, becomes less and less massive compared to the rest of the plant, and that starch gets converted into leaf and above ground tissue so that the plant can capture more energy and grow larger above ground tissues, and less of that energy gets stored as starch in the tubers that we eat. And so that's all through the sensation of temperature. So these plants are kind of measuring the amount of time that they've spent at a certain temperature and coordinating their growth of above ground tissue versus below ground tissue in accordance with that change in temperature that they're feeling. And so we could, and this group from Germany did this, actually tuned the plant down so that it accumulates that temperature signal more slowly and allows the potatoes to still grow with the tuber quality that we want as consumers of baked potatoes or potato traders, while still growing in a warmer environment. I think it was in a greenhouse up to like 84 degrees, which is pretty warm for where potatoes normally like to grow. So by changing that signal that potatoes are integrating about their environment, we can adapt the signaling system to be more suitable to our changing environment, or just generally to the needs of humanity, as opposed to the way that these plants evolved initially.
So, what are the counterbalances? Do the potatoes sense the temperature differently? Do they grow differently? Do they grow normally? I mean, are there any trade-offs?
I mean, there's always a little trade-offs, but it could be you just have to start planting the potatoes a little earlier, a little later, in order to get kind of the optimum yield from the potatoes. But I don't think they've done full field trials yet to really get to the root of where those trade-offs might exist. But that's another kind of thing that my group is particularly interested in, are some of these trade-offs that are built into the genetic program that plants have evolved with, and how we can rewire these networks to avoid some of those trade-offs. So one of the most fascinating things to me is that plants respond with two different defense pathways to insects versus microbial pathogens. And these two pathways, insects versus microbes, are mutually exclusive. So there's some evolutionary contingency there that makes, if one pathway is on, the other pathway must be off. So if a plant is challenged with both insects and microbial pathogens, which is often the case actually, right? These insects are full of microbes and maybe infecting the plants with microbes. The plants can't respond to both at the same time. And so actually, microbes and insects to some extent will take advantage of this and turn on the opposite pathway. So microbes will turn on the insect defense pathway and therefore turn off the microbe defense pathway. And so in that way, the microbes hijack the plant immune system in order to circumvent detection and circumvent the normal defense pathway. So things that as an engineer, I'm really interested in trying to figure out and be able to re-engineer some of these evolutionary, like the vestigial tail of plants, right? Things that were built in from the very beginning of the evolution of plants, that as genetic editing tools come online, we can potentially change and make plants better at doing what they're doing, because we're gaining the ability rapidly to engineer biology, and we don't just have to tinker anymore the way that evolution has done for millions of years.
I mean, the mere fact of understanding that, an attacking bug could use its microbes to attack the plant versus the bug physically attacking it. Or it could use the microbes in it, or the microbes in it could, turn off certain functions in the plant to allow the bug to munch in the leaves unimpeded. Just that alone is a much better understanding of what's going on versus thinking, oh, well, this bug eats leaves and that's the end of it.
Yeah.
It sounds like you've expanded the scope of what we understand,
Yeah, that plant, pest, and pathogen ecology is really a fascinating field. And as we're doing more and more sequencing of microbes that exist on and within plants, and kind of getting an idea of how the plants are actually detecting the really complex mixture of microbes that they're having to deal with constantly, is a fascinating field of research right now.
Do plants have an interior microbiome, or is it just exterior to them, like exterior to the root bundle? And is there a different set of microbes that live on the leaves?
Right. So there's endophytes, which are microbes that live within plants, right? Endo and phyte is plant. Endo is inside and phyte is within plants. And so depending on the plant species, you might have quite a lot of endophytes. Others, like our common model organism, Arabidopsis thaliana, does not have a lot of common endophytes, but the lactobacillus and other lactic acid bacteria that actually make pickles what they are and kimchi what they are all endophytes. These bacteria that live within the plant that we can select with really salty solutions in order to preserve the food.
Oh, so like kimchi, the bacteria that ferment kimchi are an endophyte of the kimchi plant?
Yeah, of cabbages. So they're actually living on and within the leaves of the cabbage plant that you make kimchi from.
Well, that's interesting. So they're not,
and there's
bread, I guess the yeast it comes from can be blown in on the wind or come from outside,
right.
but this comes from the inside.
Yeah, and the yeast that live on the surface of grapes that originally fermented wine is all just microbes that have figured out how to take advantage of the plants that they're constantly coming in contact with. And, there's inevitably some coevolution that's going on there. A lot of microbes have figured out how to make, or, since the microbes and plants inevitably share a common ancestor somewhere back in evolution, the pathways that plants have co-opted to be their signaling molecules, microbes share some ability to produce those signaling molecules. And so some of these microbes will actually produce plant hormones and manipulate the plant signaling systems in ways that are beneficial to them. And that's really what we try and do in the lab, is take advantage of some of that, some of these shared pathways or shared signaling mechanisms that plants and microbes have in order to break down the really complex signaling pathways that are built up in plants. And plants, for the most part, have quite large genomes. And throughout the course of plant evolution, the genomes of plants have duplicated many times. So most plant genomes are actually 3-4 duplications of one original genome. So instead of having one copy of each gene playing a separate function, you'll have 3, 6, up to 30, 40 copies of each gene, which are doing slightly different things, but they're all sharing some sequence and sharing some function. And so being able to pull out each of those 30 or 40 different copies of a single gene and look at it in isolation in a system that doesn't have the rest of those parts is really what my lab is working on, trying to relate how these genes affect function in the plant and how each of those single copies of a large gene family, is what we call these 30 or 40 copies of each gene, what each of those copies adds to that signal that the plant is trying to transmit and trying to integrate from its environment in order to appropriately adapt to the place that it's growing in.
What are the ways in which plants? Sure. What are the ways in which plants sense? Do you think they can see? Do you think they can hear? Or do they just go by chemical gradients and pressure gradients?
Yeah, so definitely chemical gradients, pressure gradients, the fluxes of these chemicals. But we do know, I'll stay away from the plant neurobiology argument. If you haven't had Simon Gilroy on, you should have him on your podcast at some point. But
okay, I will.
Plants can definitely see. They have photoreceptors. They don't have eyes like we do, but they can detect whether light is being reflected from neighboring plants or whether it's coming directly from the sun. And so in that way, a plant can detect if it's being shaded by another plant, if the light that it's getting is not of the highest energy wavelength, right? It will grow away from that spot to try and get out of the shade, or just elongate all the cells in its stem in order to try and outcompete its neighbors.
What about artificial shade versus the shade from the canopy from another plant? Has that been studied to see if there's a difference in how the plant reacts?
I'm not sure what you mean there. Like artificial shade from just a fence or something?
Right. If a plant's shaded by
Right.
leaves, we know how it reacts. But what if I just shade it with, I don't know, a piece of cardboard or something?
Yeah.
And pop it up over the plant.
Yeah.
How
So
does it react differently?
that'll definitely change how the plant's going to react, because the plant is actually seeing the full spectrum of light that's hitting its leaves. And it's detecting blue light, red light. It's reflecting back green light, right? But in the red part of the spectrum, that the plant, neighboring plant's leaves would be reflecting that red light to the plant that we're talking about here. And when it senses that red light, that's what induces that shade avoidance response. So when you have this ratio of red to far red light that's low, so when you have a high level of far red light compared to red light, that shade avoidance response is going to be induced. So,
well, again, have we
maybe
tested that that's the only mechanism? Maybe it's what if you put a green object the same color as the plant above it, but it's artificial, like a plastic plant? Would it still have the same shade avoidance, or would it have a different one? I don't know. Maybe there's more to it.
I'm not sure if that's really been done,
color.
But I think so long as the transmission spectrum looks like that through a plant canopy, that I believe it would be that way. It would still induce that same shade avoidance response. But
okay, what other
there's other mechanisms that plants use to communicate with one another. Going back to that defense pathway, when the insect defense response is activated, plants actually produce volatile compounds. And there's all sorts of volatile chemicals that plants produce to induce the defense responses of neighboring plants and say, I'm being attacked by bugs, turn on your defense responses as well. So we can imagine engineering plants so that we could prime their defenses when we knew bugs were going to be at the worst. Or we can also imagine priming development of plants, or repressing, for example, the production of flower buds on peach trees until we add some signal, give the plants some novel signal that they might not experience normally, to induce that new developmental circuit that we've rewired. So this is something that I think, as an engineer, is a possibility for improving the way that agriculture is currently done. We've had several times in the last few years huge losses in stone fruit crops because warmer winters are causing the flowers to come out earlier, and then a hard frost kills the entire crop. Whereas if we can control these signals that are causing those flowers to open, we could say, okay, this is definitely not the time of year to start flowering. Let's wait and send the plants that signal later, or tune that pathway rationally, delay that flower opening and bud production until later points in the year. And that way we can avoid that hard frost.
How would you modulate the plant's reaction to frost? I mean, it seems like a life-saving mechanism. How would you do it in such a way that it wouldn't hurt the plant, but it still could tolerate colder temperatures?
So I'm not talking about like the protection mechanisms that plants have to colder temperatures normally, but when buds have opened and then a frost happens, it'll kill those fruit that would have formed.
Okay, so what are you hoping to change the behavior then?
So if we can create a novel signal that would induce the formation of the buds, so instead of normal plant development happening, we put a repressive circuit on that bud development and wait until later in a warmer winter, till after that chance of a hard frost, and then add that new signal that would relieve that repression of bud formation and then allow the buds to develop after that chance of a hard frost is has passed.
Okay, I got you. That makes sense.
Yeah, so
what?
by delaying the development or tying some new signal to these developmental events, we could potentially do a service to agriculture.
How do you anticipate that plants are going to react to possibly higher carbon dioxide levels and global climate change?
It's an interesting question. Some plants might actually do pretty well. There's some evidence that plants increase their root system mass, which for some plants like potatoes might be a good thing. But of course, as I mentioned earlier, potatoes are pretty sensitive to temperature. So as we increase in CO2, temperature inevitably must also increase, right? So if we can adapt them to temperature, but still get that increased root biomass, that might be a good thing. But for cereal crops, right, where we're not worried about the root biomass at all, we just want seeds. The seed biomass, a lot of the evidence that I've seen is not going to increase very much, but there has been some really interesting work trying to kind of disrupt that carbon-nitrogen balance that plants are trying to maintain by adjusting the amount of root system that they have, right, where they're pulling more nitrogen from, whereas the above ground tissue, their leaves, are where they're fixing carbon. And so by disrupting that root to shoot tissue ratio, we could potentially adapt plants to be more productive in the face of climate change. So
I guess if you think about
I think there's possibility.
Horticulture in general, I mean, we've been modulating what the roots are exposed to tremendously, through fertilizers and hydroponics and all kinds of growing systems. So maybe it's not so crazy to modulate what the leaves are exposed to.
Yeah, definitely. As much nitrogen as has been applied in the past 40 years, since the middle of the Green Revolution, the amount of carbon dioxide that will be increased might not be that dramatic.
I don't know, yeah. Any other plant defense mechanisms that you're like shocked by or amazed by, or you think are really, really super cool?
Just the arms race, I think, is one of the most fascinating things. As microbes are growing, they're doubling. The microbes that we grow in the lab double. Every cell divides every 90 minutes for yeast, or every 30 minutes for most bacteria. But plant cells are dividing on a much slower rate. Plants have maybe 2 generations a year. Most of our crop plants have 1. And the ability of plants to keep up with these rapidly evolving microbes is really fascinating. So interested in the future to look a little bit more in depth at how plants are adaptable to these microbial threats.
From what I've gathered, plants are, chemical masters, and they can make all these amazing chemicals, volatiles, and, give off sap, and, chemicals in it, change the, I guess, you can call it the taste or the nutritional content, or, of their own stems. When animals eat them, they can make them poisonous to them.
I forget who said it, but somebody said that plants are the best chemical engineers. That's definitely true.
Do we know where in a plant that happens? Is it just systemically the plant's able to do that, or is there a pseudo organ or place inside the plant, or is it just inside each cell
Oh
itself,
yeah,
or they can make all these chemicals?
there's all sorts of different compartments within the plant, right? There's the all the different organelles that most of your eukaryotes have, right? The mitochondria, but then plants also have chloroplasts, and plants have these really large vacuoles and lysosomes, peroxisomes, all of these different membrane-bound compartments within their cells, in addition to really specialized cells like trichomes and some of the cells in the cuticle of the leaf that can really accumulate lots of lipids and waxes and things. So there's all of these really nice little compartments that plants have to do different chemistries, whether they need a acidic condition or a slightly basic condition, really high amounts of reactive oxygen species to do some of these chemistries. Is really fascinating how plants can get all of these different chemical reactions to be coordinated in such a way that can produce these toxic compounds or signaling compounds, or just different ways of storing energy.
Okay, interesting.
Yeah.
So what's you said you were focusing on, I guess, the redundancy of certain processes within plants, how many genes are responsible for creating certain chemicals, or can you talk a little bit about more of your focus? What are you specifically looking at?
Yeah, so we're really interested in two chemical hormones. So these hormones, just like we have hormones that control our appetite and our metabolism and our growth and our moods, plants also have these chemical hormones that are being pumped around the plant constantly and are changing in amounts depending on what they're experiencing in their environment. So we deal with one hormone called auxin, and this is really a group of chemicals that all transmit this similar signal or slightly different versions of the same signal that coordinate plant growth. And so this auxin is flowing around the plant, and in the plant stem cells, the tip of every shoot and tip of every root, there's a group of stem cells that are perceiving this auxin signal, and depending on the timing and the concentration of auxin that and coordination with other hormones, that controls the way that these stem cells grow and divide and really change their fate over time in order to produce new lateral roots or produce tubers or change from new branches forming at every the tip of every shoot to becoming flower buds, for example. And that's regulated by a lot of different hormones, but this kind of, some people would say, master plant growth hormone auxin is involved in coordinating that stem cell behavior over time in almost all of those cases. And so this signal is, doing a ton of different things for the plant, and perhaps because of all those different behaviors, or perhaps in order to produce all those different behaviors, a really complex response network, really, of all sorts of different genes within the plant are required in order to create all of those unique behaviors that we see in different plants. So it's still a big mystery in the field how this network of different genes is coordinating these different behaviors, because there's not absolute specificity for this one copy of the hormone sensor, for example, is required for forming new lateral roots. This is required to specify that a new lateral root can form from this initial stem cell. And then this gene is required in order to actually initiate the process. And these 3 genes play some role in there. If we knock each of them out, they have some effect on the timing. So, we've been doing all of this work trying to parse apart what this auxin signaling network is really able to do. And I think hopefully we're getting close to the point where we'll actually be able to rationally re-engineer this pathway in order to change plant behavior in the future. So that's kind of the broader goal of my lab is, can we gain an understanding of these parts and how they interact to coordinate this plant behavior such that we can re-engineer plant behavior. And then
what I wondered is timing in plants. Like certain fruit trees take 3 years, 5 years, however long to bear fruit, even if they appear to be full grown. How does the plant know? Okay, how does the plant count the seasons? Oh, I'm 3 years old, now I can bear fruit, or I'm 5 years old. How do you think that happens?
That inevitably is part of the molecular clock. So all plants and all organisms have some sort of circadian rhythm, right? They're detecting the amount of light that they're collecting every day and measuring the length of each day, in that way. And they're also measuring hot and cold periods. So they can.
But they must have a memory. Some kind of abacus, some kind of internal abacus or memory to know, okay, I've been around for 3 years, 3 seasons, now I'm ready to produce fruit or to bloom. Yeah.
And so I'm not very familiar with how exactly that process works on the maturation of fruit trees. But plants have some memory that's retained in one of those signaling networks. And, perhaps it's just the concentration of a certain protein is increasing every year or decreasing every year. And once it gets down to a certain level, then the plant can actually produce flowers. And so that's actually flowering locus C is a protein that does that and kind of senses as temperature decreases actually after the first year. So flowering locus C controls flowering in mostly biennial crops. So plants that will, or a lot of weeds are also biennial. So after one warm growing season, when plants start to sense a cold period, that flowering locus C protein will actually get slowly degraded over time. And once the plant has experienced enough cold weather, that flowering locus C protein will get down to a certain level that will actually relieve repression from that flowering pathway. And so you'll actually induce flowering after a cold period in that case. But maybe that mechanism, but I'm not an expert in that. So
it was funny when you talked about circadian rhythms. I realized that I don't think people appreciate how much they're like plants, or appreciate how much plants are like people. I don't know. I know it's a very general statement, but when you talk about all these plant behaviors and everything, I mean, like, when you think of a plant, you think, oh, it's critical, the day and night cycles. It's absolutely critical to the plant's functioning. But we don't think that about ourselves. Let's say, like, I'm going to go to sleep really late and work the overnight or something like that. Or, oh, it's not important for me to get up when the sun comes up.
I'll catch up on sleep later. I think there's more and more evidence that it is pretty critical to human health too.
For a plant, it's critical, but person, but maybe that's not the case. Maybe if we looked at plants and what they do, it would inform us of what to do, and vice versa.
Yeah, I think that's really very, very true. And there's a proven phenomenon, plant blindness, that if you show anyone a picture that has thousands of plants in it and has one lion, for example, and you ask people what that picture is of, say, oh, that's a lion. And no, there's actually an entire forest there. There are all of these beautiful plants, ginger flowers, whatever. They're all in this picture, but as animals, we're tied to the animal or something. But really, how many plants do we interact with every day? Are you sitting at a wooden desk? How many plants have you eaten today? We just tend to brush them off, but they're absolutely critical and very, very cool biology to study.
So what's your big hope for the next couple of years? What would you love to figure out?
I really hope that we'll be able to figure out some of those evolutionary contingencies that are built into how plants have evolved, and be able to do some proof of concept studies to show that we can remove these contingencies and improve the resistance of plants to pests, or improve the performance of crops in the future. So I really hope that we'll gain enough knowledge to be able to rationally engineer the simplest of behaviors in crops in the future. And be able to. So one of the aspects of my lab that I'm really excited about, being able to translate the knowledge that we can gain from really quantitative studies in the lab using our microbial systems, and being able to really parse apart what's going on in these plant signaling networks, being able to directly translate that to how changing genes within a plant or mixing together genes through traditional breeding can actually give rise to different traits. Using the wealth of sequencing information that we have today, if we other groups have sequenced thousands of different varieties of many different species, our model species, Arabidopsis thaliana, as well as thousands of rice genomes, hundreds, maybe 1,000 different soybean genomes, tons and tons of different corn genomes. And if we can characterize some of these critical signaling pathways and each of the different genetic changes that exist in those thousands of different genomes, we can get an understanding of what genetic changes give rise to the strongest changes in the way those signals are sent or perceived. Right, so if we can look at these different genetic changes in our microbial system and be able to characterize how they function, how they're perceiving those signals or sending those signals differently, then we can go back to those plants where those maximally different functional changes exist and figure out how that difference in signal relates to the actual difference in the plant form and function. So I think by going about things in this way, we'll hopefully be able to more rapidly engineer plants for adaptation to climate change, or rapidly evolving pests and pathogens and other challenges that plants and agriculture will face in the future.
Gotcha. Well, very good. We're just about out of time. So what's the best way for people to get in touch and ask questions and talk to you?
Sure. I'm on Twitter. My handle is @rclaywright. Feel free to reach out to me there. Be happy to answer any questions. You can also check out my website on the Virginia Tech Biological Systems Engineering or Translational Plant Sciences website.
Okay, very good. Well, Clay, thank you for coming on the podcast. I appreciate it.
Yeah, thanks so much, Richard. I appreciate the opportunity.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.